chair will be announcing Dr. Maulik Shah right after we get through some formalities with pediatric grand rounds. In the past, we have utilized different systems, but I want everyone to be aware that you do need to sign in to the high mark system. Melissa Wood from CME is in the corner of the room there and can help you sign in today if you haven't done so before. I've also linked the website on both of the boards. Please do sign in because that's how we count our attendance for grand rounds, and it's how we can continue to accredit grand rounds. Emily is passing out some paperwork. Please also, at the end of your session, please do fill in your evaluations because again, that is required for CME accreditation and it's a vital part of how we can continue to have these talks. Again, in the past, we had a time limit of 72 hours. My understanding is that that has been lifted and we do have a little bit more time to be able to complete the evaluations. But as soon as you can after the talk, please don't forget to complete your evaluation. That being said, Dr. Wood.
Thanks. So it's my great pleasure to introduce Maulik Shah, whom I consider a dear friend. He and I have spent, I don't know how many weeks of our life in Ecuador seeing patients together. So he's a great humanitarian. He volunteers his time all over the world. But he has a lot of expertise that he's going to share with us today that have to do with his training. So I want to give you a little bit of his background. He did his undergraduate degree at Virginia Commonwealth. He's a native Virginian, kind of. And then he did an MD PhD program at the University of Virginia with training at the NIH. And then he went on to Baylor to do internal medicine and genetics, both the residency and fellowship. Followed up that with a faculty appointment at WashU in St. Louis. At that point, he and his wife moved to Florida, and his wife is a medical oncologist, adult oncologist. And he started to be kind of starting his own companies, working on all kinds of big data work that he's going to share with you. And I will say, he's one of the most interesting, well-traveled, and smartest persons I've ever met.
Oh, thank you, David. Appreciate it. And if you don't know, David, I would say, is also one of the great humanists that I've met. So he ran the global health program, I think, at University of Florida that went to Ecuador. And I'm a big traveler, so I'd been to Ecuador, and I remember getting this email that said, we're looking for physicians that want to come. And I was like, Ecuador? Yeah, that's me, I'll go. And that's how I met David way back when. Little did I realize we'd end up sleeping in some pretty remote areas, sharing outhouses. And so let me tell you, you really get to know someone when you go through that. So I was trying to give a talk today that really focused more on some of my clinical activities rather than a lot of things that I do from the corporate standpoint. But if anyone's ever interested in knowing about having a non-traditional path through medicine, I can tell you a lot about that. So the secondary title of this is Establishing a Telegenetics Clinical Genomics Program, and I'm going to hopefully demonstrate to you why this is beneficial, and we'll go over some of the difficulties in interpreting genetic data and genomic data for the most part. So because this is a CME lecture, I have to give you my disclosures. So I have a lot of financial disclosures. So Genetic Medicine Clinic is a private genetics clinic that I own, which I'll talk about a little bit, that provides these genetic services in addition to partnering with University of Central Florida and University of Florida. I own or I'm the medical director of Applied Ingenuity Diagnostics, which uses artificial intelligence to analyze rich genomic data for the most part. I'll mention a little bit, I'm the head of research for a German company called Numaris, which does metabolomic testing. And I have adjunct faculty appointments at a number of places. I was telling David, I also have a consulting company, and through that, I'm an expert in writing SBIR grants, if you're not aware of those. Those are small business institutional research grants. So I have or own about 12 or 14 different companies where I license technology from universities, and then we use government-based grants to essentially move the technology forward to the point where we can reach commercialization by raising angel capital or venture capital for the most part. So I do a whole variety of different things. I'm happy to talk to you about any of those later on if you want. Our objectives for this specific lecture are just going to talk about models of telegenetics. As you're probably well aware, genetics and genomics is increasing at an exponential rate, and the problem is there just aren't enough trained individuals on the back-end genetic side to provide care or even provide consultation, and hence we came up with this telegenetics program. And then we're going to talk about different categories of genetic tests and interpretation of genetic tests. So our clinic really is different. So I know you guys have a clinical geneticist. We don't supplant the clinical geneticist. That's not our goal at all for the most part. We're a combination that has the clinical genetics piece, but we also have the testing piece that blends it together, and the research piece, and then we live in that little bubble in the middle. And I'm going to show you how that's advantageous in a variety of cases. So I'm the director. A lot of our genetic counselors come from the University of Central Florida. And our goal is to build a comprehensive telegenetic service with clinical laboratory services, really to deal with those patients with unexplained genetic diseases. So if you remember, the title of this talk is Telegenetics and the Variant of Unknown Significance. And if you haven't seen a variant of unknown significance, as you start doing genetic testing, you will see more and more reports where the test result is not obvious, right? It's easy if they give you Fragile X testing and they give it to you, or they give you Rett. But more and more, you're going to see genetic test results that require interpretation, and require interpretation in light of the clinical phenotype. And that's where we come in a variety of different cases, from cancer predisposition, cardiovascular disorder, a variety of pediatric disorders. The reason this is really relevant is genetic technology and genomics is increasing at a tremendous rate. So the number of clinical genetic tests that are available is more than 2,000 now, right? So if I asked most of you, you guys are probably aware of like Fragile X, Rett, you could probably name off 4 or 5, but the reality is there's more than 2,000 genetic tests that are clinically available, meaning you could go and order it for your patient right now. The question is, are you ordering the right test for the right patient, and then are you interpreting it correctly? In fact, there's a lot of data that shows that approximately 25% of all requests for complex genetic tests were changed following review by a geneticist or practitioner. And it's because, I'll give you a hypothetical. Let's say you have a child with autism, or let's say global developmental delay. What test do you order, right? Do you start off with whole exome sequencing? Do you start off with Fragile X? Do you start off with chromosomes? Do you do a chromosome microarray? Right? Really, all of those are possible, and so the question becomes, what's the right test? And the danger is ordering the test that actually doesn't answer the question that you need answered, right? Which is why the child has whatever the child has. And increasingly, and these numbers have actually gotten worse, that there's a lot of children out there that are getting tests that were not clinically indicated and were misinterpreted for the most part. So to give you an example, when, not in this program, but we went to an adult home that had individuals with a variety of developmental delay. Interesting enough, about 40% of them had some type of genetic testing done that was not diagnostic. Of that 40%, we did a clinical evaluation, and the geneticist ordered the appropriate test, and about half of them we were able to make a diagnosis that was not defined. And these are individuals that had been in a home for quite a while. So one of the difficulties is that the amount of, the pace of technology is just growing much faster than we can keep up with the educational piece to understand what's going on. The other issue becomes, as you'll see later, how to interpret these test results. So The number, indications, and complexity of genetic testing is increasing, and it's going to continue to increase even more. So it's not surprising that mistakes are made, and this results in failure and delays in getting the appropriate test results, communication of results that are incorrect. Just a couple weeks ago, we saw a young girl with global developmental delay where the doctor had ordered a chromosome microarray. It was normal, and so they said we weren't sure what to do, and it was clearly obvious that the child had the phenotype of Rett syndrome, and it took one quick visit to order the correct test and refer the patient. Otherwise, the poor girl had not been getting physical therapy, speech therapy, or anything else based on having an incorrect test and incorrect interpretation. Our approach is multidisciplinary. So obviously we have genetics, but we also have genetic counselors, and the pediatrician is a key component in this because you're the one in this kind of remote cloud-based process that we have. You're the one that's hands-on with the patient, right? We can't really do the exam, and so we need a lot of assistance and guidance from the primary care who's going to provide a lot of the phenotypic data as well as some of the coordination in terms of data gathering. So a big problem or issue always is collecting the appropriate data. So as you know, many of these children, especially with rare disorders, you're probably the third or fourth person to see them, right? They've usually bounced around, had various tests done at different places, and those tests may be important in the overall evaluation. And so we need someone to kind of coordinate as a case manager all of that. So we have a genetic counselor, a medical geneticist, and then we have a molecular pathologist who is someone that is kind of trained to help interpret and analyze the genetic test results. We provide consultation to all healthcare providers needing further information on genetic testing. So in Florida, we work a lot with OBs who are getting non-invasive prenatal testing. I think I mentioned, or maybe I didn't in my disclosure, so I'm on the board of Natera, which is one of the largest companies that does non-invasive prenatal testing. So non-invasive prenatal testing is really supplanting the triple screen for the most part in high-risk women. It's part of the guidelines from the American College of OBGYN. But once again, the question becomes, if you know something about NIPT, and we'll talk about interpretation, you end up with lots of variants of unknown significance, right? So you end up with a genetic SNP analysis that you're not sure what to do with, and in that case, you have limited time to provide some information to the family so they can make decisions regarding that pregnancy. So we can also just simply review the test orders, make sure the correct test is being done, right? So, sometimes you may want a chromosome microarray, sometimes you may want whole exome testing. And then the goal in this is we don't want to become like this big cloud-based, genetics clinic. Our goal is to, through this process, provide education back so that essentially these kind of processes can occur internally within your group. So, once again, for some things there should be protocols for genetic testing that aren't always there. Like if you've got global developmental delay, yes, you should be doing a chromosome microarray and Fragile X, but you shouldn't necessarily stop there, right? So what's the next step after you get that? It depends on if it's a boy, a girl, what age. Are we talking, global developmental delay is kind of this big catch-all, but there's a lot of phenotype data that can help direct what the next appropriate testing should be. And so for a variety of conditions that you're seeing in pediatrics, we can assist in terms of figuring out what the appropriate process should be. So just to give you an idea of the workflow, essentially you send a test requisition, not for the genetic testing, but just that a consultation is required. We have a bunch of people then obviously review it, work on collecting the appropriate information. We contact you, the pediatrician, to once again, most importantly, identify what's the real question that you're trying to answer. Are you trying to make a diagnosis? Are you trying to get genetic testing for the purpose of management? The actual question you want answered is the integral part to what's going on. So we try to make sure we communicate to make sure that happens. Then we have people that do back-end insurance authorization. So most of the time we don't get paid on the clinical part, and that's fine. But if more advanced testing needs to be done, and especially with genetic testing, you'll almost need some form of insurance authorization previously, especially because a lot of this genetic testing is expensive. One of the advantages we have is I also own the company that does the genetic testing, so we make money on lots of other things, so we can eat the cost for a lot of these things without worrying too much about it. It goes into our workflow, and the way genetic technologies are going, the cost to do things is coming down tremendously. It's really the personnel and other part to it. But we still try to get insurance authorization, and we help in that process. A lot of that requires having appropriate letters of medical necessity, and this is where we once again work with the pediatrician to put together the appropriate letter for the insurance company to give appropriate authorization so we can move forward. And then, you know, you guys are collecting usually the specimen, whether it's saliva, what it is. We actually, in this process, we ask for consent for biobanking, especially in rare disease. One of the issues I always have as a geneticist is trying to make any genotype-phenotype correlations when the data set is so small, right? If there's only 10 patients in the whole world ever with a certain genotype, it's hard for me to tell a parent what to expect for that child based on a data set, right? It's not like hypertension where, we have a data set of, millions where I can say, yeah, if your blood pressure is high, this is probably what's going to happen to you for the most part. So when we're dealing with rare disease, that's one of the greatest difficulties and challenges, and that's the area where patients really want to know, and their families, what the outcome might be. So we try to biobank if they're willing for it, because then that can be used for subsequent analysis down the road. It also allows us to enroll that child, for example, in clinical trials if necessary later on. So we have a child just last week that got enrolled in a brand new CRISPR trial for muscular dystrophy, but they were looking for a very specific version of muscular dystrophy based on the genetic alteration, because they knew the technology works only there. And so they were having a tough time finding patients, because once again, they didn't need patients with muscular dystrophy. They needed patients with this genetic alteration to really test the technology, and this allowed us to help participate for it. So once again, we provide the genetic analysis, and this is the core part, which is the data analysis part. So if you order genetic testing from a number of my colleagues that also own genetic testing labs, they will give you a nice formal report that's very good. But the difference is they're giving you a report just like when you order a CBC, right? The lab doesn't know anything about your patient, isn't really interpreting that test for you. You're required to do it. In this case, because we know some of the genotype information, the phenotype information, we can make sure we're doing the appropriate data analysis to get the results that you want, for example. And I'll give you an example of that in just a moment. And then obviously you get reports. Since we own, or have since we have that genetic information, many times we don't end up with an appropriate diagnosis, or we're left in still a state of uncertainty. But this allows us to re-examine that child, at least on the laboratory standpoint, at a later time point, because the phenotype may change, especially in children, greatly in 1 year or whatever time frame. So in 6 months later, if you say, hey, by the way, the child developed, severe nausea and vomiting, intractable, whatever, we can say, oh, that changes the picture a little bit. Let's go back and reanalyze the data. So I'm going to give you a case example just to show this. So this was a 6-week-old child transferred to the University of Florida for altered mental status, respiratory failure, hypoglycemia. Child was extremely hypothermic, cyanotic, acidotic with a lactic acid. So obviously, child went to ICU, had a lot of supportive care done. Originally, the question was, is this an inborn error of metabolism? Is this a fatty acid oxidation disorder? And going into all that, I mean, they did all the correct things. And it turns out that, no, it was not. And so they were left with the fact that this child had a severe cardiomyopathy with left ventricular non-compaction, and they weren't sure why. So we went through the process and we elected, instead of just doing a cardiomyopathy panel, because this child had a somewhat unusual case picture, we decided to jump to whole exome sequencing. So if you're not familiar with whole exome sequencing, our exome is really the genes that are active. So meaning they're the genes expressed at any given time point, right? So we have a lot of genes that are quiescent, or they may be expressed only in whatever stage of development. So at any given time, if I draw my blood, right, the exome is the genes that are active. So I'm essentially taking all of the RNA, converting it back to DNA and sequencing it. So I'm not looking at the whole genome per se, I'm looking at a much more limited subset. But the reality is that subset is really about 14,000 genes that I'm looking at. But by looking at the RNA converted back to DNA, I still am able to essentially sequence and get and identify genetic alterations. So in this child, what happened is he was found to have a variant of unknown significance in the myosin heavy chain 7 gene. And then he had a second mutation in the troponin T gene. And it was also a variant of unknown significance. So what is a variant of unknown significance? It just means that it's a variant and it's never been reported before, so we don't know what the heck it means. And you're going to see this very, very frequently. I imagine you're going to see this more often than you actually see a real test result, because you're going to do more and more genetic testing. And as we do more and more genetic testing, the databases that exist are going to be updated, but they're not updated yet. So like this child had 2 variants of unknown significance. So this has lots of importance for the child, right? So one, we have to figure out, do these mean something? So both are associated with some type of heart phenotype. So the easy answer would be sure, absolutely, right? They're both associated and make sense. But when you have variants of unknown significance, at least in children, you have to figure out, are the test results normal? Are they really pathogenic? If they're variants of unknown significance, what they are, or are they just incidental finding? So in genetics, a true normal is not that you didn't find a genetic alteration. A true normal is you didn't find a genetic alteration, or you found a genetic alteration that's been associated with a normal phenotype, right? So the absence of finding something doesn't necessarily mean that the test result is normal. Similarly, a pathogenic mutation is not necessarily pathogenic, right, unless it's been reported to alter the function of the gene and been reported to result in the phenotype somewhere else down the line, right? So you might just be measuring the variant that separates, me from anyone else. And then you're going to get lots of variants of unknown significance. And I won't go into that today, but one of the big issues is you end up with a lot of incidental findings. So when you're looking at the whole exome, right, I may find out that the patient is a carrier for CF. I may find out that the patient has a genetic alteration in BRCA and is at a high risk for breast cancer. For a young boy, that probably isn't relevant at this moment, but it might be relevant later on, right? What happens if you find out that the child has an incidental finding of Huntington's disease, right? Now you've opened up a can of worms that you weren't necessarily supposed to, but it has implications of the family, right? So one of the big issues with genetic information is information once learned can't be unlearned, right? So once you know it, you can't do it. So one of the most famous cases is a set of identical sisters whose mother had breast cancer at a young age, and one sister sued the other because one sister wanted to do the genetic testing and one did not. And she made the argument, well, if you get the genetic testing, I find out information that I necessarily don't want to know for the most part. So genetics opens up a lot of issues in terms of counseling for these patients. So once again, a normal is that there's no clinically significant changes identified in the DNA of that specimen for the gene that you're specifically looking at. The difficulty, once again, may be that you may be limited by the test methodology. So maybe gene sequencing is not the correct answer, right? So for example, Prader-Willi, which you're probably all familiar, is a disorder of methylation. So if you do whole exome sequencing on a patient with Prader-Willi, guess what? The test results are going to be normal. but you're not going to identify or diagnose Prader-Willi because you ordered the wrong test for that. And just because you get a test report that says the whole exome was normal doesn't mean that the child had that because you ordered the wrong test. Similarly, the things that are caused by microdeletion syndromes or other things are going to come back essentially normal on a whole exome. So just because you get a normal report doesn't necessarily mean it's normal. It's normal if the genes that you were particularly interested in or think correlate with the disease phenotype are normal for the most part. Once again, pathogenic has to be pathogenic only because, once again, the genetic alteration is there and it's been identified and associated not only to be a pathogenic mutation, but associated with whatever the phenotype that you're looking at. And we talked about that. So when you get a variant of unknown significance, this is where we kind of assist or help because you have to spend a lot more time going into the next steps. So you have to figure out for this child who has these two variants of unknown significance and two genes, both associated with cardiac phenotypes, which one is responsible? Is it both? Is it one? Right? Because that is going to make a big difference to this family. They need to know because they might want to have children in the future and know their risks. It makes a big difference for this child. Are they going to be eligible, for, let's say, for a heart transplant or not? How are you going to manage them long term? I'm sure these parents are thinking, you know, what will my child be able to do? Will we get out of the hospital? Will he play soccer? Right? You have to know these kind of things. Who else in the family needs to be tested? So these are the questions that come up. And so the first thing we did is we said we'd like to know the parent's status in this. So we drew samples on both parents to identify, was this de novo, meaning these genetic alterations were just identified in the child, or were they inherited at all? We weren't sure, obviously. So when you have a VUS, usually you test the parents, and if neither parent has the VUS, then that helps because then you can say this is a de novo mutation. You still have to go forward. You always have to worry about non-paternity, right? Anyone know what the non-paternity rate in the U.S. is? Any idea? Anyone? It's about 10%. Look around you, right? So if you don't look very much like your father, there might be a reason for that. For most. And this goes across culture and demographics, et cetera. There's lots of studies in the U.S. So the non-paternity rate is about 10%. And so that's not insignificant also, though, from our standpoint when we're interpreting these test results, that a normal, once again, doesn't necessarily mean normal. And then, once again, it depends on if the parent has it and if the parent has a phenotype that's associated with it. So for this child, we got a multi-generational pedigree. There were no unexplained deaths in the family, especially of children in infancy. There's no history of cardiomyopathy, and no real history of heart disease. They were pretty young. I think the parents were in their 20s for the most part, so they weren't quite old. The myosin heavy gene, interesting enough, when we tested a parent, was identified in the father, right? So the plot thickens now. Okay, so he's got this, but once again, he doesn't have a phenotype. So we said, let's get an echo on him. So we got an echo, and he had a normal EF, normal heart volumes, no real issue. Although interesting enough, for a very young guy, he had right bundle branch block on EKG, which is kind of a unusual finding. It can be there as both an idiopathic finding in young adults, but that was the only thing. And then, interesting enough, the mother had the troponin T mutation. She had a normal echo, an EKG, and once again, no unexplained deaths in the family. So now we're faced with interpretation. What does this mean now? So clearly the child got one gene from each, but neither of the parents had a cardiac phenotype. And so what does that mean? So we have to know something about these genes. So it turns out that the myosin heavy chain gene, or the myosin 7 heavy chain gene, is associated more with hypertrophic cardiomyopathy and right-sided cardiomyopathy. So this graphic shows all the genes associated with cardiomyopathy. It's actually much more expansive. Interestingly enough, when you look at cardiomyopathy gene associations, there's a lot of overlap for the most part. So when you go the opposite way and you take people, let's say, with dilated cardiomyopathy and you do genetic testing on them, and find out, you find a lot of associations, right? So it's never a single, it's not a situation where there's a single gene associated with a single phenotype. And the reason for this is, once again, what we're talking about is what we call complex genetics, not single gene disorders. So if we take something like PKU or galactosemia, those are single gene disorders. So 100% burden of disease is responsible for that, is based on that gene. In contrast, if I take something like hypertension or type 2 diabetes, that's a complex phenomenon, right? You haven't heard about a gene for hypertension because there isn't just one. It's interplay of a lot of different genes associated with that. And so once again, you see that there's a lot of overlap. But interestingly enough, myosin heavy chain gene is more associated with hypertrophic cardiomyopathy. It's also associated with cardiac fiber disproportion, which is a very specific separate phenotype that you can see in children. It's associated with sudden cardiac death for the most part. So it's been reported with hypertrophic cardiomyopathy, both idiopathic and familial. So there are families reported where with HCM where MYH7 mutations track specifically down it. Similarly in dilated cardiomyopathy, there's actually families reported where MYH7 mutations track with the phenotype in multi-generation families like a single gene disorder. It's associated with kind of a rare muscular dystrophy, and it's associated with left ventricular non-compaction, which is part of the phenotype that our child had. So once again, the question becomes, does this child have a abnormality in myosin heavy chain 7 contributing to his phenotype or not? So in addition to just knowing a little bit about the gene, nowadays you can go one step further to try to do genotype-phenotype correlations. You can look at animal studies, transgenic models, etc. So interesting enough, if you look at animal studies, the myosin heavy chain gene is predominantly expressed in fetal life. It's expressed a lot less later on, and it can change according to stress and hypothyroidism. But one of the most important things is mutations in this gene impact contractile properties, hence the association with cardiomyopathy. Interesting enough, there is a correlation. So when we went back and we looked at the familial cases, and this hadn't been reported in the literature, it turns out that most of the familial cases, the genetic alterations were at the head of the protein, so the ones that tracked. Our patient had a change that was much more distal in exon 8 or 9. And we found that the more acidic the amino acid change was in these families, the more severe phenotype. So this required quite a lot of digging in Medline to come up with these papers, read them pretty thoroughly to try to come up with these correlations to find out what's going on. And the reason I tell you this is because our child had, I forgot what it was, but it did have a change, or the predicted change based on the alteration was from a neutral protein to a more acidic protein. And our child had a mutation somewhere here. And interesting enough, this is, we went back and looked at all the structure-function relationships from all the different reports there were. And one of the things that's hard, I think, to see, but you see a lot of dilated cardiomyopathy associated with alterations over here, where hypertrophic cardiomyopathy was associated more at the head of the protein. Interesting enough, we found a number of reports where, once again, people had looked at patients with left ventricular non-compaction and went backwards and essentially had found MYH7 mutations. Once again, not sure if they were pathogenic in those patients, but that's what was reported. And we found there was at least 7 different MYH7 mutations, but interesting enough, they all clustered close to exon 8 and 9, which is similar to where our patient had genetic alteration. But I will point out the genetic alterations weren't just limited to this gene when we went back and looked. So if you look at patients with left ventricular non-compaction, they track with a variety of genes, but just breaking it down to the ones with MYH7, that's what we found. And then when we look at the troponin T, so I think you guys are all probably aware of troponin T. It has a lot of different isoforms. It has varied expression in cardiac muscle, But interesting enough, it also has expression in skeletal muscle and fast fiber, right? So why do I tell you this? Remember, the mother had this genetic alteration. So knowing that, this may have some impact now on mom. Even though she had a normal echo, right, et cetera, it doesn't mean that she may not have some problems later on. So a lot of the genetic alterations, right, can have reduced penetrance, meaning you can have the phenotype much later. And they're only in their 20s. So there's still a risk that they could develop a phenotype in their 30s or 40s or whatever it may be. So this also is associated with all different forms of cardiomyopathy, and I won't go into extensive detail about it. We found one case report where there was a 20-year-old with non-compaction who had a mutation that was very close to the site of our proband. We started looking at the literature to do some genotype-phenotype correlations, and people have done a variety of things where they've created transgenic animals and they've knocked out different parts of this. And the functional studies show that especially if you knock out troponin T near where our patient had the alterations, you usually get alteration in myocardiac contractility. And interesting enough, in the families where troponin T tracked with cardiomyopathy, you could see all of the different phenotypes in that family, meaning you had some patients with hypertrophic cardiomyopathy, you had some patients with dilated cardiomyopathy in that family, and some patients with right-sided cardiomyopathy. The transgenic mice, though, also showed left ventricular non-compaction, okay? And then this was kind of interesting. So digging really deep, we found a paper that reported a patient from Iran. And if you look at this mom right here, she actually had a mutation in troponin T and myosin heavy chain 7, and she had a severe dilated cardiomyopathy. So interesting enough, in her family, she had a whole bunch of unexplained deaths, although they didn't mention in this article how old those kids were or if they were adults when they died. And in this case, there were 2 other individuals with severe cardiomyopathy that also had inheritance of both genes. So this is obviously different from our family because our child got it, one gene from each parent. But regardless, when you have both of these genes together, you can see a significant feature of cardiomyopathy. So once again, now that I've given you this knowledge, you can see that we had to go quite a bit more in depth to make really some correlations for the child. So what are the risks in future pregnancies? So the risks, I think, are a little bit complicated to really figure out. If I was going to counsel this patient, I would say, once again, kind of autosomal recessive risks, because you could have another child that also inherits both for having a very severe phenotype or cardiomyopathy. But I think because both these genes are associated with cardiomyopathy and the parents are young and they could have a reduced penetrance for the most part, meaning they just haven't manifested, that there's still a risk that they could have a child with cardiomyopathy. I didn't mention, or maybe I did, that the myosin 7 gene was also associated with sudden cardiac death, right? And so there's a lot of reports where it alters the electrophysiology of the heart. Once again, if you remember, I told you Dad had a right bundle branch block, so this may have bearing on him. Now, right bundle branch isn't necessarily associated with severe electrophysiologic abnormalities and sudden cardiac death, but I just find it pretty coincidental and interesting to find an unusual cardiac finding like that in that. It may have, obviously clinical impact on the outcome of their son. He actually did fairly well, so he was put on a left ventricular assist device for a number of weeks and then underwent heart transplant. He had a minor stroke in the middle of all of that. He's discharged from the hospital. He has some developmental delay, but he's still young and the ultimate phenotype isn't quite known. For me, for this family, for management of adults, I would probably recommend that each of them get an echo done every year. I might even recommend an EP study on the dad to make sure, because of this risk of sudden cardiac death. And then both parents have siblings that had not had any children yet. So this affords us the opportunity to screen their siblings to give them some guidance on what the next steps may be. So one of the difficulties, as you can see, when you get a variant of unknown significance is going the next steps to really figure out if there's genotype-phenotype correlations. Because if you don't do that, you really couldn't answer all of those questions that the family has. And this is the area that's going to become very much more difficult with genetic technologies coming to the forefront. So I see whole exome sequencing all the time, and pretty much in every report, there's variants of unknown significance, right? Because many of those are just identifying the variability between two individuals. It doesn't mean that they're pathognomonic. This is where we really need good phenotype information from the pediatrician, and then we have to put the big picture together the best that we can. Can I still say absolutely that it was this combination that does that? No. But the fact that I found a case report that had severe cardiomyopathy, the fact that these tracked with left ventricular non-compaction, which was part of the phenotype, I think there's a lot of inferential data that we can use to help this family make associations for it. So just to give you an idea, when we put this program in, we like to study anything, everything. So we looked at how referrals were made and triage, et cetera. And you can see when we had patients with rare disorders or unexplained phenotypes that this is after having gone at least one round of whatever genetic testing was indicated by whoever the primary was. A fair number of kids still end up for secondary referral, and many of them end up with positive genetic testing after appropriate testing is done for the most part. So it shows you the advantage of having this type of referral service available, especially for many of you that may be in rural areas and not have access to a geneticist. One of the big advantages we have, because we have a large number of genetic counselors, is at UF, the waiting list to get into genetics is about 8 months, 9 months for the most part. Most of Central Florida is like that. So when I practiced as an adult geneticist, I had a waiting list more than a year because there's no adult geneticist. I was like the only adult geneticist between Atlanta and Miami for the most part. So one of the problems is there's just not enough trained individuals. So we put this program in place to kind of expedite that process, because especially in peds, you don't necessarily have 8 months to wait for that appointment to get going. I mean, the reality is you have to start that testing process as quickly as possible, and it doesn't require that much to have a little bit of direction to go in the appropriate path. And then depending on what the test results, we can come up with that. And sorry, this slide got cut off from a PDF, but we did an evaluation of how useful is telegenetics, meaning did people like the technology? Could no one figure out the technology? Did they think they were getting what they were going? And all the data is over there that you can't see, but the results were all positive. So most patients felt that they got the appropriate level of care as if they were seeing someone in person. I think a big part of that was the fact that the pediatrician was involved intimately in the process and the care. The technology is set up so you don't need to have an office or whatever. Most of these consultations were done with the patient over their cell phone or over their laptop. The way the technology works is we essentially send them a link, they click on it, and boom, they're online with our genetic counselors at that moment. And so it's a very simple process. It's actually built on a HIPAA compliant background, so there's no issues from the technology standpoint. And I think that's it that I have. Thank you very much. I'll take questions. Oh, yes.
So one quick question for you. With rare disorders, it seems like much of the way that we have traditionally solved problems like that is to group rare things together to further be able to study it. Can you comment on how hard it is from a regulatory and ethics perspective for storing potentially powerful genetic information for patients for the future? Is there a lot of regulatory oversight in that? Should there be, or what do you think on that subject?
Oh, that's a loaded question. Thanks, John. I appreciate it. It's hard to answer that. So I mentioned this in the last talk. So during the Obama administration, there was something called the Genetic Information Non-Discrimination Act that was passed. That prevents genetic information to be used against you from the perspective of healthcare portability, right? So the fact that you have genetic data that says you're at high risk for Alzheimer's, for example, cannot be used against you to prevent you from getting into a nursing home, from the insurance perspective. In contrast, things that are financial products like life insurance, et cetera, is allowed to use that. So there was actually a nursing home in Florida that asked for genetic testing for all patients entering, and it's currently going under tort law, and the case hasn't been resolved to understand whether that's ethical or not. So yes, I think at some point governments need to come together to figure out what genetic information should be protected and to what degree for what purpose, for the most part. When you're talking about rare disorders, like what we're talking about here, I mean, we try to consent everyone if we can for what we do, but the issue becomes there is still a possibility of loss of that information out into the great wide world. I think it was a much worrier risk than it is now, only because 23andMe and a number of companies have gotten out there, Ancestry.com, et cetera. I don't know if anyone's done that. But if you have, what you probably didn't realize is most of you checked a box that said they can take your data and do whatever they want with it. And so your data is probably already out there to a certain degree. So the reason 23andMe costs so little, actually, is they eat the money on the technology, right? So for them to do that chip, it actually costs way more than the $89 that they charge you for it. Where they're making money is on the data. So they turn around and sell that data for a whole variety of different things. And I mean, a lot of that data is available on public databases. So if you've seen recently, there's been a whole bunch of cases where criminals were identified based on their genetic profile, not of people that were in the database, but doing public database searches and finding individuals that were somewhat related nearby and making appropriate correlations.
Do you think it would make sense to have the SNP genotyping from 23andMe be available to primary care physicians? I mean, I went through that, and it seemed to have some useful information, but it's not like I could go to my primary care physician right now and say, I've got SNP,
Yeah.
What should I do?
Yeah.
Do you think we should move in that direction?
Well, what we're going to move towards is having a technology that helps physicians interpret that. So like you said, there's a lot of valuable data in there, but there's a lot of crap in there too, right? So SNPs, there's, what is that last count? There's about 4 million SNPs. So SNPs are single nucleotide polymorphisms, so they represent the difference between me and Emily right there, right? So the difference between us is only 0.1% of our DNA, which is represented in all those SNPs. So you're really identifying the variability between one individual or other. But when I get patients that come with that data, the problem is it's hard to know what to do with that data. Some of it is pretty easy. So you can look at like the CF carrier status, and I think everyone here could counsel them appropriately, or the Tay-Sachs carrier status. But there's a lot of SNP data in there that just raw data that can be used for a variety of things that 23andMe doesn't report to you, right, that you could do. The problem is, unless you know where to go and what to look, then it's difficult. So we actually have kind of computer-based AI algorithms that will search through that data to provide actionable plans for you. The question becomes, once again, do you want to know information that you didn't ask for the most part? Like, since you did it, and I told you I could go in there and tell you if you're at risk for Huntington's disease, would you want to know? Okay. Would everyone want to know? How many people would not want to know? Yeah, see, so that's where the difficulty becomes. I can extract a lot of information there, and it's the blueprint for who you are. It doesn't mean you're going to get that phenotype, but you have to use it quite judiciously for the most part. But I think it's going to be built into EHRs eventually. There's going to be enough genetic information technology, just like labs come into the EHR and you get your CBC. There's going to be enough raw data that eventually comes where EHRs will build in clinical decision support tools into the process. So that data is taken. And I actually own a company that does that. So yes, I know it's going to happen.
Just as a follow-on to Dr. Schweitzer's question, I noticed in your example you used primary medical literature to augment your interpretation, so you found a case report in the family. What are you doing to ensure that that information, for example, this patient is now available for someone who uses a different company to
Yeah, it's a great question. So the question is really, how is this information now disseminated so it's not necessarily a variant of unknown significance moving forward for everyone? So there's a number of online-based databases now that track mutations. So for example, there's a cardiomyopathy project, and all genetic testing labs, if they're worth their salt anyway, start reporting their positive results in there with whatever phenotype data they have. And so now, in fact, before we report that as a variant of unknown significance, right, the algorithm searches through that database before it's even reported as a VUS. We even go further now in our reports where we do a gene ontogeny evaluation. So I didn't go through all that, but it turns out that you can track these genes, obviously from the evolution standpoint. So if it turns out that the genetic alteration is in an area that's highly conserved all the way down, to a mouse to a human, then that's probably pretty important. It doesn't tolerate mutations in that area for the most part. So if you have alterations in there, they're probably bad. In contrast, if it turns out that that's an area that's highly variable for the most part between lots of different species, then that you have to interpret and say, well, there could be some flexibility in terms of really calling this a pathogenic variant. So we go through a lot of different things, but we're all reporting back in. The problem is most of those databases are usually set up through academic partnerships. So like the cardiomyopathy one happens we just set up at Washington University in St. Louis. There's ones for Tay-Sachs, there's ones for just about everything. But at some point, someone's going to have to make an effort to have a centralized database of those. That's a great question. Anyone else? All right, well, thank you very much. I appreciate your time.